Hi everyone, welcome to my presentation. My name is Stephen Kotiang, a graduate student at Wichita State University. The title of my presentation is Probabilistic Factor Graph Modeling and Analysis of Biological Networks. In this work, we present a computational framework that combines the formulation of probabilistic graphical modeling, standard statistical estimation, and integration of high-throughput biological data to explore the global behavior of biological systems. We formalized our model as a probabilistic factor graph representation of biological networks. We then tested our approach on DNA microarray data corresponding to two well-researched gene networks in E. coli. The objectives of this research include to find the state of a gene with respect to the state of other genes interacting with it in the network and formulate this relation mathematically. The state could be the expression level of a gene. In addition, to quantify how well the existing experimentally verified gene regulatory networks are explained by the biological data. A simple but effective model people use to model gene networks is the Bayesian graph, as shown in this figure of 4 nodes. We show genes as nodes and interactions as directed edges. A gene influences the action of another gene through creation of some specific proteins. Also, altering the state of one gene can cause other genes interacting with it to change their states. In discrete terms, each gene can take a value from a range of k finite logical states. A state is defined as a range of gene expression levels given by the statistics of the discretization model used. We assume that a gene takes a value j in k with a probability j, where pj is referred to as a marginal probability. Our wish, therefore, is to study the regulatory relations among genes. Such relations, for example, determine the level of gene expression for a gene from a set of genes or transcription factors interacting with it. We may ask ourselves, why take a probabilistic approach? In practice, the information available for regulatory relations is incomplete and variable certainty. You may have experimental data, but they only provide partial information on biological systems, and this motivates the employment of a probabilistic methodology for inferring such relations. In this slide, I describe our probabilistic model. We transform the gene network on the left into an equivalent factor graph on the right. A factor graph is a bipartite graph that associates variable nodes in circles on one side of the graph and factor nodes in squares on another side of the graph. In doing this, the factor nodes represent the regulatory functions of a gene. These functions denote the belief that the gene takes a certain state with respect to an assignment from its parents. For instance, F1 is the regulatory function over gene G1 and is controlled by gene G2. In probabilistic terms, the factor graph represents the joint probability distribution of the network as a product of local conditional probability distributions given by this equation, where Z is nothing but the normalization constant, and PAIs are the states of the parents. The general workflow of our model is described as follows. We obtain uniformly normalized experimental data and gene network structure carefully curated from the literature. In this sense, the network and the data become our model inputs. We then apply gene-specific discretization to the continuous data using mixtures of Gaussian model. Once you have this, you can apply maximum likelihood estimation to learn the regulatory functions. However, since biological systems are highly cyclic in nature, we perform marginal inference on the model using loopy belief propagation to find the probability of a gene being in a particular state. I will explain the loopy belief propagation in the next couple of slides. Then with optimal marginals, we then perform statistical analysis. In message passing algorithms, nodes pass and collect information about themselves from neighboring nodes as shown in this figure, and then they compute their beliefs. We use belief propagation because they are considered simple, tractable, powerful message passing algorithms, and they are widely used in the engineering fields. LBP is a variant of message passing algorithms. It is an approximate inference algorithm and is known to produce good results when it converges. In this figure, the messages are described as follows. The lambda messages are messages from the child nodes to the parent nodes, while the mu messages are messages from the parent nodes to the child nodes, which are as well described by these two equations. Then the belief update equation is also given as shown. That at the end of each and every iteration, each and every variable node computes its belief. This process is then repeated iteratively in time until a stable configuration is achieved. Then at convergence, the converged beliefs are the marginals of the variable nodes. We then tested our probabilistic model on E. coli SOS response network and acid regulatory networks as shown in figures A and B respectively. The SOS pathway regulates and coordinates cell survival and repair after extensive DNA damage. Similarly, the acid-resistant pathway enables the organism to survive in extreme acidic environments. We obtained our data from the M3D database. This compendium data consists of 466 sample points obtained under various experimental conditions. Then for computational efficiency, we discretize the data into 2 and 3 logical states of discretization for analysis. We then applied our methodology to the data given the network models. We ran our method to find the steady-state level of gene expressions. Then we compared our results with observed data from the experiment by computing the Pearson correlation coefficients. Figures A and B show the correlation plots of the SOS response network for both 2 states and 3 states levels of discretization. In both plots, we see that the coefficients of correlation are well above 0.96, indicating a good approximation of the observed states by our probabilistic model. Similarly, For the acid resistance regulatory network, the correlation plots are also shown. Here too, the coefficient of correlation are above 0.90, thereby again indicating good approximation of the observed states using our probabilistic model. Using these plots, we demonstrate the performance of our probabilistic model through a single gene deletion, that is, gene knockout analysis. Gene knockout is one of the ways to test the robustness of our model to the existing data. In the gene data, we identify REC-A gene as the most knocked out gene in the experimental samples, having a total of 68 observation points, that is in the SOS response pathway. We then modified our model and applied the inference algorithm to estimate the steady states. Figures A and B show the correlation plots of the SOS response network for both 2 and 3 states discretization levels respectively. Here too, we see a correlation of more than 0.9. Furthermore, we tested the consistency between the considered networks and their respective experimental data. We did this by comparing the performance of our model on these two networks against their corresponding random networks. We implemented a perturbation that depicts permutation of nodes on a graph to create random networks. In this perturbation, we end up with an identical topology to the original network, but the gene expression profiles are perturbed. Figures A and B show the average correlation plots between the inferred marginals and the observed experimental states with increasing number of randomized nodes. In general, with increased randomization, the average correlation coefficients decrease. We deduce that random gene regulatory networks are inconsistent too and do not adequately explain the gene expression profiles. In summary, we explored a probabilistic factor graph representation of biological networks and applied message passing inference algorithm to investigate the allowable stable states of biological networks. In addition, we tested the consistency and robustness of our method in predicting steady state of genes against experiment data. These findings reveal that the state-of-the-art gene regulatory networks considered are strongly supported by the experimental data. That marks the end of my presentation. Thank you for listening. This slide provides the reference to our publication. Thank you.